Hello students, today I am going to start with another very important cellular organelle that called Golgi Bodies or Golgi Apparatus. Okay, the name Golgi Apparatus is said when Golgi Bodies is taken into consideration with Fascicles. Together, Golgi Body plus Fascicles is known as Golgi Apparatus. Okay, the name Golgi Body, as you can see, was discovered by Camillo Golgi in the year 1898. So the name was given by his name only, Golgi Bodies. He was a scientist who discovered Golgi Body in the year 1898. Okay, Golgi Body is an important organelle of eukaryotic cell. It is found in all eukaryotic cell, that is plant cell and animal cell. It is a single layer organ, no double layering. It is a single layer organ, as you can see in the diagram in the screen here below. Let me zoom in for you. This is the Golgi Bodies. You can see it is marked with very dark black in color. These are the Golgi Bodies or Golgi Bodies, however you pronounce, doesn't matter. Okay, so in my last video of this chapter, Cell the Unit of Life, in part 2, we have seen about endoplasmic reticulum, its structure and function. Remember? Yes, please, if you have not seen the last video, please have a look and then come to this video. Okay, we have seen how ribosomes are attached to endoplasmic reticulum. When ribosomes are attached to endoplasmic reticulum, it is called as rough endoplasmic reticulum. Rough endoplasmic reticulum are those endoplasmic reticulum where ribosomes are attached, and this ribosome makes protein and passes it to the lumen or luminal space of endoplasmic reticulum for further processing. I have already discussed. Yes, ribosomes makes the protein, and this protein is transferred to the lumen of endoplasmic reticulum for further processing. Ribosomes just makes the protein, but that protein is not in usable form. So to make it usable and for further refining, this protein that is made by ribosome is transferred from the surface to the luminal space inside endoplasmic reticulum, and this processing is called N-linked glycosylation. N-linked glycosylation, the slide I will be showing in the last video. I'll show you the process actually. Let me just explain a little bit of N-linked glycosylation that occurs in the processing of protein in rough endoplasmic reticulum. Let me just show you it. Yes. Yes. See, N-linked glycosylation is the attachment of sugar molecule, carbohydrate molecule, that is oligosaccharide known as glycan, to a nitrogen atom. Okay, in endoplasmic reticulum, N-linked glycosylation occurs, and when this protein is transferred to Golgi bodies, there O-linked glycosylation occur. You don't need to go very detail about this and glycosylation and all. I'll just explain a little bit of it, but it would have been more comfortable if it was a classroom. I could have explained it a little bit more in an easy way, but then it's okay. Just remember that in endoplasmic reticulum it undergoes N-linked glycosylation, in which a specific carbohydrate is joined to the protein chain. That carbohydrate that is joined, that glycan or oligosaccharide that is joined to the protein chain, this is a type of signal which specifies where the protein is to be sent. Who needs this protein? It's just like a what to say, just like a seal, or just like something like that is meant for only specific organ. If I have to, for example, if I have to send this protein to lysosome, then this is only targeted for lysosome. This protein won't go anywhere to plasma membrane or to other cell. No, it will only go to lysosome. This is called N-link glycosylation that occur in the processing of protein in rough endoplasmic reticulum. So N-link glycosylation means a specific carbohydrate group or sugar group or oligosaccharide is linked to protein chain. This sugar group, which is added to protein, is to specify the exact or the desired place to where the protein is to be used or to be sent, right? Okay. After the processing of protein in endoplasmic reticulum, it is further sent to Golgi body through package body called fascicles. Okay, you can see small round-like structure from endoplasmic reticulum. Here you see the arrow mark. There, yes. Here you see the arrow mark from endoplasmic reticulum. It is sent to Golgi body by the formation of a small round structure, bud-like structure that was at first attached to endoplasmic reticulum, and then it detaches from endoplasmic reticulum, and then it moves to Golgi body through the cis phase. What is this cis phase? Now tell me. Cis phase. You see here? Yes, cis phase. Cis phase is that side of Golgi body which is facing the endoplasmic reticulum. Yes, through cis phase, the Golgi body receives the protein inside vesicle for further processing. It need to be further processed. In endoplasmic reticulum, it was not modified that much, so it need to be modified in the Golgi body. Okay, through package body called fascicle. So it is received by Golgi body through cis phase. Okay, the protein received by Golgi body through cis phase, or cis phase is also called as entry point, then undergoes further processing and then final modification in the cisternae of Golgi body. You remember in the last video I have talked about cisternae. What is this cisternae? Yes, see here, cisternae. Here in Golgi body, you see the cisternae are flattened tubular structure and are not attached to one another. They are just singularly parallel to each other. Okay. The cisternae has two phases, you see, cis phase, and this is the trans phase. Trans phase is also called as exit point or exit site through which the protein, after modification in the Golgi body, is excreted out of the Golgi body to the targeted or the desired place after modification. Remember, Golgi body modifies the protein. Suppose if some organelle needs protein to be made into glycoprotein, then this is done in Golgi body. If lipid that is received from smooth endoplasmic reticulum, remember, smooth endoplasmic reticulum is involved in the making of lipid. Yes, if this lipid is transferred to Golgi body through smooth endoplasmic reticulum, Golgi body further modifies it into glycolipid. Suppose, say, if some organelle is suppose I am the owner, I am ordering you, please make me, don't give me lipid, give me glycolipid. So what will you do? Suppose you are Golgi body, you will modify the lipid into glycolipid, and then through vesicle you will send it to me in a package. Vesicle is just a packaged body. It just packages the protein to be delivered, safely delivered to the targeted organ. Okay, am I fine? Okay. Then the cisternae has two phases, as I have already told you. Cis phase, side facing endoplasmic reticulum, which receives protein from endoplasmic reticulum, and the trans phase, or the exit point through which protein moves out of Golgi body after modification. Okay, fine. Okay, this protein after modification is transported to desired organelle or targeted place by getting packed in vesicles. Yes, these are this round structure, you see this round structure, these are all vesicles that are broken out, that are detached from the Golgi body itself in the formation of a bird-like structure that detaches themselves from the Golgi body. They pack the protein inside them and they move out of the cell or to some specific organ which requires not organs, organelle that requires the protein. Okay, so this is the work of Golgi body. In short, in summary, I have told you the whole story of what happens in Golgi body and endoplasmic reticulum. Let us go to the next slide and see in detail what actually happens. Here I have just given the summary of everything. Just have a look at it. Okay, here in the left corner side I have given you a little bit of their functions of Golgi body. Their function is transportation. Yes, they are transporting the protein, they are modifying the protein, and they are packaging the protein in the vesicles. Okay, what do they package? They package materials like protein, lipids, carbohydrate, etc. Anything which is received by them through the cis phase, they modify it and package it through the trans phase and then transport. Okay, let us go to the next slide. Yes, here you see the Golgi apparatus, also called as Golgi body or Golgi complex, is an organelle found in most eukaryotic cell. Yes, most eukaryotic cell. It was identified in 1898 by the Italian physician Camillo Golgi and was named after him. Yes, after he discovered this organelle, he gave it the name by his name only, Golgi body or Golgi apparatus. Apparatus it is called when it is combined with vesicle. Okay, the primary function of Golgi apparatus is to process and package macromolecules such as proteins and lipid after their synthesis. Yes, it is synthesized and then it is transferred. It is modified and then it is transferred. It is particularly important in the processing of protein for secretion. The Golgi apparatus forms a part of the cellular endomembrane system. Yes, in the part 2 of this chapter I have told that endomembrane system consists of Golgi body, endomembrane, endoplasmic reticulum, nuclear envelope and vesicles. The Golgi is composed of stacks of membrane bound structure known as cisternae. Yes, this is also what I have discussed just now. Now, let us go to the next video and show you the detailed picture of what is happening. That will give you a clear picture. Okay, see here, let me rotate. Yes, you see the cis phase is receiving the protein through transport vehicle, that is the vesicle. Cis phase receives the protein that is not fully modified, so cis phase receives it to the cisternae of the cis phase and then it transfers it or to the target place to the trans phase. This secretory vesicle that is packaging the protein and then transferring it to the target place. If it is to be if it's to be sent to the organelle inside the cell like lysosome, cytosol, then it transfer or যদি এটা যদি প্রোটিনটা যদি বাইরে দিতে কোনোখানে সেন্ড করতে through the plasma membrane, then also it does the same thing. Okay, see, rough endoplasmic reticulum is sending the protein that is packaged in vesicle, little bit modified. Further modification goes on in Golgi apparatus and then it is sent through the plasma membrane or inside the cell. Okay, am I clear? Please pause the video and have a clear look at this diagram. You will get a detailed knowledge of everything what I have said. Please excuse me if I am too fast. Just pause the video and try to catch what I am saying. Okay, now let us see the function of Golgi apparatus one by one. As I've already said, now let me be very specific. Function of Golgi apparatus: number 1, storage, modification, packaging of product in vesicle. Yes, this is what I have said just now. Formation of complex sugar from simple sugar. Just just like formation of glycolipids from lipids, right? Okay, third point, secretion is the main function of Golgi complex. Yes, secretory proteins and lipids are packed and released on the surface by exocytosis. Yes, through the transphase, it is exit from the Golgi body. Fourth point, it helps in the formation of cell plate during cell division. Yes, in plant cell you have seen that during cell division means division এক cell দিয়ে দুই সেল ওয়ার যখন দুই সেল হইতো এক cell plate is formed between two cells. That you will learn in botany portion. You will learn this about. Just remember, as of now, just remember this. Okay, the fifth point, the Golgi apparatus is also involved in the formation of lysosome and peroxisome. Yes, so to some extent it forms lysosome and peroxisome. Lysosome will take it in the next video, or maybe in the next video. Lysosome are called suicide bag of the cell. We'll touch about this in the next slide. So these are the very specific function of Golgi apparatus. Please learn it, remember it, try to understand and memorize it for the exam point of view, and also just know everything about it. Okay, this is again a clear picture of Golgi apparatus. This is the cis phase, see the trans phase, newly forming vesicle that is formed from the body of Golgi body, secretory vesicle that carries the protein, lumen of Golgi body where the protein is modified, cis phase, cisternae, incoming transport vesicle that is coming from endoplasmic reticulum. Just pause the video again and have a look. Okay, the next slide, or then the last slide, I have already told you about N-link glycosylation. So have a look at it once again. Okay, before I say about Golgi body, in endoplasmic reticulum we have seen N-link glycosylation. What is N-link? N-link is nothing, actually it's just site and where nitrogen site in arginine is a nitrogen where the protein carbohydrate is attached, so N-link glycosylation occurs. And yeah, in Golgi body O-link glycosylation occurs. Just remember, in Golgi body O-linked glycosylation occurs, and in endoplasmic reticulum N-link glycosylation occurs. Okay, thank you.